Hello, everyone. Welcome back. I'm on today with evolutionary biologist Dr. P.Z. Myers, and we're going to talk about uh, embryology, evolutionary developmental biology, and other topics. I hope, and it should be a whole lot of fun. So, tell them a little bit about yourself, Dr. Myers.
Oh, I've got to work. What? Oh, okay. Yeah. So I'm a biologist. I'm at the University of Minnesota Morris, which is a small but very lovely liberal arts college up here in the Great North. Um, I'm actually more of a developmental biologist than an evolutionary biologist. We can talk about that in a moment. And I came into evolutionary biology by way of development because, as you know, Evo-Devo is kind of a big thing. And more developmental biologists are recognizing the importance of evolution in their work. And I think it's reciprocated. I think a lot of evolutionary biologists recognize that development is kind of a critical component of evolution. So that's where my interests lie. I'm at a teaching university, so my primary obligations are not research, but education. But I do dabble in zebrafish on the side. And I got a nice little colony of fish producing embryos for me right now, just across the street over there.
So, what do you look for in particular with the zebrafish?
Well, my primary interests are in cellular behavior. So how do cells recognize where they are in the embryo, pattern formation and things like that. And how do they know where to migrate in particular? Because animal cells have this cool property of being able to migrate within the embryo. So what we like to do is observe the movements of populations of cells and see if we can figure out any rules behind their behavior.
Do you have? Are you aware of the swapping of or in the inversions in the protostome and deuterostome ancestor?
Oh, well, yes, that's the definition of protostome and deuterostome is they use different poles for different parts of the body.
So,
So yeah.
so do you think it was the simply a migrating of the nervous tissue, or do you think it was an inversion, like a genetic inversion?
Uh, well, no, it had to be a complete flip,
Okay.
because not only are the pores different. So in deuterostomes like us, the closure of the blastopore forms the anus, and in protostomes it forms the mouth. But also what you find when you look at those is we, for instance, have a dorsal nerve cord back here. This is where our nervous tissue is. And if you are looking at an insect or a crustacean or any kind of arthropod, nervous system is on this side. Mm-hmm. So they're flipped upside down. And that's been known for. Geoffrey St. Hilaire made a big deal about this in about 1803, 1805, somewhere around there. And he actually made an early argument that what they were inverted forms of each other. He had a somewhat tortured process by which this happened, and he actually argued that the vertebra is the core of the animal. And what happened is we have our vertebrae on the inside with our nervous system on top of it, and insects have the vertebrae on the outside and the ventral nervous system. No, that doesn't work. The cuticle is not homologous to vertebrae, but other parts of his theory were kind of interesting. And as we got more and more molecular evidence, it became ever clearer that that's what was going on.
So, there were two inversionary events. There was the one at Nephrozoa, and then the one at Deuterostomia between the ambulacraria and the chordates.
Right. So again, we don't know what the ancestral animal looked like, right? Mm. So it's not really clear exactly what was going on there. That we could imagine that the ancestral animal was ambiguous in its orientation, that there wasn't much of a difference between dorsal and ventral, and that it was, just as it was bilaterally symmetrically, it was dorsal ventrally symmetrical, and so that it was free to rotate whatever way it wanted. That's one model for it. Another is that there are some interesting suggestions that, for instance, there are different fibrous tissues ventrally and dorsally in the vertebrate. And the argument is, well, they were both set up the same way, but in vertebrates, the dorsal one became the nervous tissue, and in the protostomes, the ventral one became nervous tissue. So it wasn't really actually a physical rotation so much as different sides of the animals developed differentially.
That's really cool. But it would have been a bilaterian, yeah.
Yeah. Oh, yes.
So why did the radial symmetry reappear, or is there any explanation as to why radial symmetry reappeared in echinoderms?
Oh, is there an explanation? Ooh, well,
Or a possible explanation, I guess.
yeah. I mean, for instance, if you look at something like a starfish or a sand dollar, they're radially symmetrical, all right, but the embryos are bilaterally symmetrical.
Okay.
And that's kind of what counts. And so we think that they were ancestrally bilaterally symmetrical. And then the radial symmetry emerged as some kind of possibly an adaptation to their environment, their particular lifestyle. I don't know. Or it could be that it's a side effect of something else that occurred.
Mm-hmm.
Yeah.
That's really cool. And so you mentioned zebrafish earlier, and so zebrafish are one of what you call model organisms, right?
Yes. So there's a number of model organisms out there. Model organisms, they have their strengths and their weaknesses. Their strength is that we tend to pick animals for models that are simple, develop rapidly, have good genetics, or easily bred in the lab. These are all things that are advantages when you're working in the lab. But they also have disadvantages in that they tend to be extremely highly specialized. So zebrafish, for instance, when you look at them, they're just this kind of generic looking fish. I think they're gorgeous, but a lot of people think they're kind of bland because they just got bluish black stripes on a silvery body. So they're not exactly spectacular, but they live in harsh environments on the Indian subcontinent, and they breed really, really fast. Apparently, their secret to success is that they take advantage of the monsoons and thrive in those yearly rainstorms. So they grow rapidly, they breed quickly, spew out a bunch of babies that then are ready by the time the next monsoon rolls around. So that's kind of a specialized strategy for living. And then in addition, when you've got simple systems like that, they tend to be they tend to have quirks. They tend to be focused in certain ways. So for instance, zebrafish do not have any oral teeth. So if you want to study tooth development, you don't want to work with zebrafish. They do have the pharyngeal teeth, little nubbins back there, but otherwise, no teeth. If you're interested in digestive system development, they're kind of quirky because zebrafish do not have a stomach. Esophagus goes straight into the intestines. So I know some people who study gut development in them, and there's some interesting stuff going on. I mean, there's still universalities, like they have an enteric nervous system that's just like ours, so they're good to study that way. But yeah, they all have these. They're all kind of stripped down versions of other organisms. It's like if you wanted to study cars and you decide, well, Cadillacs are too expensive, so I'll study this dune buggy over here. It's easier to work with. It's cheaper. maybe things are more accessible, but you can't pretend that you're going to be able to study, for instance, automatic transmissions or fancy stuff inside of a real car. So it's good for getting the basics, and that's why we focus on model systems. Zebrafish are just cute. They're new. People have been working on Drosophila for over 100 years.
White mice, or the little mice and frogs and whatnot.
C. elegans has been. How long has C. elegans been used?
Oh yeah, oh yeah, that's right. The what is it called? The apoptosis genes. They like
Oh,
look
yeah.
at that, don't they?
Yeah, C. elegans has been really nice for studying genetics of things like cell death and cell lineage and stuff like that. And they've been around since the 60s or 70s, as used as model organisms. Zebrafish, I know exactly when they started. It was like 1975 when George Streisinger first started working with them.
I was half expecting you to say because I was the first one.
No, I was like the third or fourth or fifth one.
Okay.
But I was in that first generation. So I worked at the University of Oregon. I actually worked in George Streisinger's lab. He's the guy who started it all. And then I moved into Chuck Kimmel's lab, and he's the guy who's sort of taken it over after that. And there was a time when I was working on zebrafish, and I think there were 5 of us total around the world.
Mm-hmm.
And almost all of them were in Oregon. So it was, yeah, it was a very small group. But it was really successful, and I think in large part because with zebrafish, the focus at first, thanks to George Streisinger, was not on development or evolution. It was on genetics. Let's just work out the genetics of these.
Mm-hmm.
Let's figure out how to do crosses. In particular, let's figure out how to clone these fish, how to generate diploids and haploids using some simple techniques, how to do artificial fertilization. He worked out all those techniques, which made it really useful for genetics. And nowadays, if you want to work on a model organism, that's first requirement, is you better have the genetic tools to probe into it. And so zebrafish got that boost right at the very beginning. It's problematic for some organisms. So, for example, another animal that lots of people use in the lab is the frog, right?
Mm-hmm.
Species of frogs, and they have lousy genetics. They're just poorly characterized. There was a guy who was doing some work trying to make an initial genetic map of them a while back. I don't know what came of that, but frogs haven't been very successful genetically, and that's kind of limited the progress you can make with them, because you really want to be able to take the animal apart at the genetic molecular level in order to figure out what's going on in the modern day. And so zebrafish have that, frogs don't. Flies have that. Flies, oh man, flies, everyone works on flies. Flies are just the most excellent model system out there because of all the things you can do with them. And second most excellent, I would say, has to be the mouse. Mice are harder to work with. I rejected them right away because, hey, internal fertilization, how are you going to get at the embryos? But yeah, so mice are second, and then I'd say C. elegans, and then would I put zebrafish in 4th place? Maybe around 4th places. The most excellent model system to work with. I recently went to a developmental biology meeting in Minneapolis, and one of the surprises for me was, I made my initial perusal through the program to see, oh, well, where are all the zebrafish talks? And nobody mentioned it. And then you sit down in the talk, and they're doing all their work on zebrafish. They just don't think it's newsworthy enough to mention in the abstract anymore. So what? Everyone works on zebrafish now. So, but it was kind of a surprise.
Oh, that's funny. Have you seen the work on zebrafish behavior, or have you seen any of that?
I have a student who is supposed to be doing that. I don't know if he's doing it. I haven't heard from him in a while. He's took away a bunch of fish for the summer, and he's going to do some simple behavior analysis. But yeah, I've seen some of that, and I've seen some of the work on learning and behavior in zebrafish.
I'm amused by results of putting ethanol in their little dishes, and then they do kind of funny stuff. I
Yeah.
think it's interesting.
Yeah, I've done some work with ethanol studies, but I'm using it as a teratogen. So I throw it on embryos and mess up their development. It's kind of a rough model for fetal alcohol syndrome. Um, Again, this is where you start discovering that model systems may not be the best, is because zebrafish, it turns out, are remarkably tough. They can handle so much alcohol that we could not possibly do. So, what's a blood level alcohol for being drunk, legally drunk? It's um,
It's like point oh something or point oh.
something. Yeah, I've
Uh,
had to worry about it, so I don't know, but I know it's really low.
yeah.
Uh, Zebrafish can handle a blood alcohol level of 3%.
Wow.
Yeah, which would pickle a human being. They would. But zebrafish are somehow able to metabolize alcohol much more rapidly than we can, and can cope with it better than we can.
Wow, that's very interesting. I've never heard of that.
Yeah, it made the teratology studies kind of difficult and at first unbelievable, because we kept jacking up the alcohol concentration, and they didn't care. They were fine. They were fine. And it was only when we started getting up around 3 to 5% alcohol that suddenly we started seeing major birth defects in our fish. Uh, yeah, it was kind of unreal.
BJ Price, a commenter, asked, "But are they happy?"
What makes a zebrafish happy? I don't know. These zebrafish are well fed and cared for. They get nice tanks where we carefully monitor the water chemistry. I'd say they're pretty happy. Um, the ones that we treat with teratogenic levels of alcohol, I don't know, maybe they are, but one common phenotype is one eye in the middle of the forehead, and I don't know if they're happy. Is Leela happy? Maybe, maybe not.
That's funny. Glad we got to talk about model organisms. I think that's very interesting because I've seen you've done some work on model organisms. I saw a paper. I think you authored comparing the fly and mouse homeobox genes.
Oh, was this the Nature article? There's a
I might have been.
Yeah, that wasn't original research on my part, but it was. Uh, I did some teaching stuff, some educational works that were published in Nature, and one of those was about comparing fly Hox genes and mouse Hox genes, and there are differences and similarities. The similarities are kind of interesting and revealing. The differences are also actually kind of interesting and revealing.
Such as, or what difference, or what differences would you consider revealing?
Well, start with the similarities. The first similarities are, of course, that they're collinear, which
Mhm.
means that when you look in the chromosome, you see genes here, 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 in a little cluster, and they're in that same order of expression in the body. So this is the front, this is the back. It's
Mhm.
all being done in the same pattern uh, as the map in the chromosome to the map of their expression in the body, which is really kind of cool. Um, this was first observed in flies by Ed Lewis and Christiana Nusslein-Volhard and Eric Wieschaus. And then it's also been seen in mice. It's also present in zebrafish. Lots of organisms show this pattern. So there's this common thing where it looks like on the chromosome, there is a map of the body plan. Nobody expected this, but there it is. Okay, so that's a similarity. They both have that same pattern. Difference, though, is that in vertebrates, we don't just have one map, we have four of them. So we have four copies of the Hox genes.
Mhm.
They're on different chromosomes, but they've got roughly the same constituents, and they have overlapping zones of expression. So what it means is that patterning in vertebrates is a little messier because, you know, there's a region here. It's not just one chromosome, one Hox chromosome. It's a couple of Hox chromosomes with overlapping domains of expression. So you can imagine kind of a combinatorial scheme for patterning the embryo. Another difference is that vertebrates have added a couple of Hox genes. They've added a couple of new ones at the back end, and they correlate with the tail. Because vertebrates have a tail, arthropods do not. And the tail is an extension of the body axis, and so they've got their own Hox genes. Other things that are kind of cute about it is it seems to get recruited, the Hox system gets recruited in other systems where you've got to lay down a linear set of coordinates. So they're expressed in your limbs, for instance. Hox A and Hox D genes are expressed in a pattern down the length of your arm and hand, and they specify position as well. Insects don't have that because limbs are primitive and ancestral in insects. But I guess when vertebrates evolved new limbs, you know, it was a matter of just co-opting, recruiting, and reusing genes they already had to set up a pattern along those limbs. And the Hox genes were convenient that way.
Like the PITX gene?
Right. So there's lots of examples of genes being co-opted and reused in development.
Yeah, I read Neil Shubin's Your Inner Fish.
Yes.
Like 2 years ago, I think it was. Very interesting book. I think that was one of the things he talked about because he showed the different, like the lepidotrichia in like a zebrafish, and then the like the fin of a lungfish, which is slightly more like a tetrapod, and then they just kind of kept, and then there was Tiktaalik and Acanthostega, and it just kind of kept going.
Yes.
And so
these patterns are seen all over the place of this reuse of genes to do similar sorts of things. And yeah, there's obviously there's differences between a fish fin and this.
What? Unless you're in southern Louisiana, in which case there might be some similarities.
Oh, yeah. Well, I grew up in Washington State. It's just as wet. So yeah, you could say the webbing is yeah, so there's differences, and it seems to be differences in terminal recruitment of genetic pathways to establish digits in vertebrates.
Yeah, there was also a gene I came across. Also, actually, it's kind of funny because there was, as you caused me to look it up in a way. I saw a Ray Comfort's movie, Evolution versus God, and he asked, and he loves the Gish Gallop, and so he talked to you a few times, or he's got a few clips of you, I should say, in the movie, and he asks you, you know, just give me one piece of evidence, and so you explained to him the stickleback, and like how they've convergently speciated, and also and so I read about sticklebacks and how some of them have lost they have lost the spines,
right,
because they're getting like grabbed by some kind of insect, wasn't it?
Yeah, things like dragonfly nymphs and so forth.
Yeah.
There's this interesting pattern going on because if you're sort of a deep water stickleback, you want to be spiky because you want to dissuade fish from eating you. Whereas if you've got lots of spines and you're in shallow water and you're down near the bottom, then those spines become sticks that an insect can grab onto and hold on while he nibbles on you. So they tend to shed those sort of things. They're also expensive because it requires lots of minerals to build up these spiky protrusions.
This is like was it ectodisplacin?
Yeah. So there's a whole series of transformations that go on in these sticklebacks. Sticklebacks, by the way, are another good model system because what's powerful about them that makes them different than zebrafish is that we have diverse natural populations of sticklebacks. So if you're interested in classical evolutionary studies, where you can look at different populations, look at population genetics, look for the basis of differences in different populations. Sticklebacks are great. Zebrafish suck, because all the zebrafish we have are lab reared. They've been lab reared for over 100 years, and so they're kind of inbred, and we don't have a lot of diversity. There have been a few expeditions out to India and Nepal to collect native zebrafish, but it's tricky to do because there's all kinds of limitations on bringing wild animals into the country.
Makes sense, yeah.
Because they can carry parasites and diseases and so forth. So there's an extensive quarantine and all kinds of procedures you have to go through. It costs buckets of money to bring in these foreign fish. And it's all because down there in Louisiana and Florida and all that, that's where they raise zebrafish commercially.
Yeah,
and
yeah.
Big industry. And they don't want some foreign zebrafish disease from Nepal getting loose in the big ponds in Florida and killing all their zebrafish.
Well, there are a lot of fish in Florida that shouldn't be there because, and not just that, but mammals, reptiles, amphibians, birds. Because people just, you buy like a little foot-long ball python at the pet store, and you think, oh, it's not going to grow to like 4 feet, or larger, and so then it gets too large, and they say, oh, well, we'll just drop it off at our local pond. And then you've seen the pictures of like these things eat alligators,
yeah,
or something like that. It's insane.
Yeah, we don't have that worry up here in Minnesota. Yeah, our winters kill off things like those anacondas and alligators, and they also would kill zebrafish. So no, they're not running loose around here. We've got other invasive species, but not those.
It was while I was looking up the sticklebacks, I also came across a very interesting fish, which I think you talked about. Actually, it might have been from one of your lectures. You talked about the glycoproteins in ice fish, the convergent evolution, and you talked about that was really neat.
Yeah, a good source for that is if you read Sean Carroll's book, The Making of the Fittest. He talks about these fish quite a bit. Ice fish are kind of cool because they're from, actually the family that they're from is spread all over the world, but they've just sort of taken over Antarctica. And the reason is that it used to be that Antarctica was warmer, that the continent was in there, and there was that nice little finger from South America pointing down. It used to connect with Antarctica, and then there was a separation between the two. It allowed currents to flow, and these currents then did this around Antarctica. They just kind of circle instead of swooping up to the equator and back down. So it got colder and colder and colder. It killed all the other fish. But ice fish acquired a bunch of adaptations that allowed them to thrive. And one is a natural antifreeze. So their blood actually contains an antifreeze that suppresses the formation of ice crystals. Another is a subtle one, and that is microtubules break down in us at cold temperatures. So a lot of fish were just simply sterilized because you couldn't carry out mitosis and meiosis and things like that in really cold temperatures. And the ice fish got around that. And then another adaptation is that as it got really cold, one of the things they did is they ditched the red blood cells. Just got rid of the red. Yeah. So that's why they're called ice fishes, because they look like little chunks of ice. They're transparent. You can see right through them. They don't have red blood. They have kind of watery, pale blood. And the reason they did that is it's a trade-off. You don't need the red blood cells because it's cold enough that oxygen saturation is pretty good in those tissues. And so it can carry enough oxygen to supply them, especially at the low metabolic rates they're operating at. And also, at really cold temperatures, the blood gets really thick. and doesn't flow very well if you're full of red blood cells. So it's a way to make it less viscous and get better circulation. So yeah, they've got a whole set of adaptations that allow them to thrive down there. Again,
Yeah,
read Sean Carroll's Making the Fittest. It's got a really
making
good.
of the fittest. All right, now you got me. I gotta look up another making of the fittest.
Yes,
Okay, all right.
I'm giving you your reading list here, and it's
We actually, funny because you just reminded me of this. We actually just got. I was emailed about it, or at least I saw my email just last week. I don't know when it was emailed to me. That we got a developmental course at LSU now.
oh,
Unfortunately, I can't take it. It's a senior course. I tried signing up for mammalogy at the behest of the mammologist I intern
uh-huh.
with, or I guess whichever preposition goes there. Uh, and yeah, he said you should take the mammalogy course. You probably do pretty well. And so I tried signing up for it, but they told me, sorry, you can't. It's a senior course, so no bueno,
Oh,
which
oh.
is kind of sad for me.
How far along are you? What year are you in?
I'm going into my sophomore year, but I
Okay.
spent this whole year. I've always been into zoology and evolutionary biology, and
Uh-huh.
by always I mean actually always.
Yeah.
I on some level I've always been interested in this stuff. I loved dinosaurs when I was a kid. I'm actually wearing a dinosaur shirt right now.
Very
Uh,
nice.
yeah, actually, let's see. There you go, camera. I had it focused on you. There you go. And so it was very interesting. I was half expecting people to be out protesting the dinosaurs, but fortunately there were none. I did
Uh-huh.
see one woman with a "Hey, I'm a Christian" shirt while I was in there, as odd.
Oh.
But anyway, but yeah. So I intern at the LSU Museum of Zoology
Uh-huh.
during the school year. Well, maybe if FAFSA gives me my work study, then I can get paid for it, which would be
Yeah,
nice.
that's always
Um,
good.
yeah, and so uh, met some really interesting people. Listened to some really interesting lectures. I even got to talk at one of our events. Uh, I'm on the mammalogy team, so it's two geneticists. I think we have a new one now. A guy just joined us from some other university, and then the guy who heads it all, who's also a professor. And so, and then there was also a senior who was working about a work study last semester, but she's gone now. And so, mostly they had me cataloging specimens. Tulane orphaned up a large part of their collection to us,
Uh-huh.
which was inhuman amounts of Ondatra. So many muskrats. They were just pages and pages and pages of Ondatra. I don't know what was going on over
Uh-huh.
there. Um, but we also got some really intriguing things, like they had a dolphin or something like that, and some other odd specimens. But yeah, I got the task of going going on an Excel sheet. All right, cabinet 8, drawer 1, cabinet Pipistrellus, cabinet 8, drawer 1, Myotis, cabinet 8, drawer 1. Yeah. So that was me for months and months. Nowhere near done.
Yeah, that's what museums do. It's kind of essential. So yeah, it has to be done.
I enjoyed it
Uh,
though. It gave me a lot of time to watch YouTube videos, some of which were yours. I
Uh-huh.
listened to some of your lectures. Um,
Uh-huh.
but yeah, funny. One of the funniest things that happened to me was is it Erethizon? Is one of the porcupines. Um, and so we had to move them basically from the bottom floor of the museum up to the top floor. And so we were shifting everything around, getting it all compacted before we moved it. And so one of my geneticist friends, he's like, sets the tray with the porcupine. He says, "Can you bring?" He says, "Can you bring it over to like cabinet 7?" And I went over and looked, and I was like, "We can't fit a whole nother cabinet in there." And then I realized, oh, he wants me to carry the porcupine. So for about a minute, it was me going, how, how do I grab this thing?
Oh yeah.
I told my girlfriend about that, and she laughed so hard.
Uh-huh. Yes, when I was a young kid, that we often found porcupine roadkill,
Mhm.
and that's actually where I got my start. digging into biology is there's a dead porcupine out there. I wonder what makes it tick. Let's take it apart.
Yeah.
And yeah, they're not easy to handle.
Even when they're dead, they still poke you.
That's right. Yeah. So I
Yeah.
mean, that was much fun. And they also roadkill tends to get kind of putrid. But
Yeah. One of the one day I was going to class and I found as I was walking there, I had to go past there's a big bell tower on campus, and so I was walking past the bell tower, and there's this dead bird that was just lying there, and it had just died. Maybe it had flown into the wall or something, but it had just died. And so, me being me, I took a picture of it for this app I have on my phone called iNaturalist.
oh yeah, I've got that. Yeah.
Yeah, and so I took a picture of it, sent and within like a few hours, one of the ornithologists at the museum had sent me, "Oh, that's a cedar waxwing. Did you get it?"
Uh-huh.
And I was like, "No, I was late to class. I did not pick up the cedar waxwing and transport it
Oh,
to the museum."
yeah. We should plug that program here. I mean, iNaturalist, it's a really nice app that,
It is. Yeah.
yeah, it lets you take a picture of something with your cell phone and then it uploads it and it makes preliminary guesses about what it is and other people can comment on it. So it invites naturalists
Hmm.
to tell you exactly what you're looking at. So
I
it's a
wish. Yeah, I wish
go
I'd
ahead.
actually had it earlier that day because our no, it wasn't earlier that day. It was I think it was like a week before or something like that. It was before I'd had it. I was walking out of the dorm area where I live, and I was like, I kicked up something, and it landed on the back of my leg. And I turned around and looked at it, and it was like a little terrestrial flatworm because it had like the kind of hammerhead
Oh,
on it, or the little semicircular head. And I said, "Oh, that's nice." Just kind of set him down. I was like, "Ew,
yeah,
got dirt on my leg now." He was
yeah,
probably just like, "What the heck, man?"
yeah, and I need it because, uh, contrary to the stereotype of biologists, I cannot step outside and classify everything in sight. I knew biologists like that, and they were awesome. You go on a hike with them, and they give you the
Hmm.
Latin names for everything. No, I can't do that because my thing is to take it apart and look at its cells.
Hmm.
If you ask me about the histology of that thing, give me a knife. I can tell you all about the histology. But yeah, that level of macrobiology, that's not
Where
my
were
thing.
you when in the botany section of my class? I should have come to you.
Oh, well, not so much of a botanist either.
Well, xylem and phloem and all that, the
Yeah,
the sinks and everything, fun stuff.
I know generally that part. I actually graduated from a zoology. Are you? Is it officially a zoology program you're in? Or
Oh no, I am a biology. I'm getting a bachelor in biology
okay,
right now. Yeah.
yeah. Back in the old days, you could specialize right from the beginning, and I was trained in the zoology department. And so, yeah, we never looked at plants,
Mhm.
which is kind of a gap in my background. But anyway, we just focused right away on animals, and all we studied was animals. It was kind of disgraceful in some ways. Nowadays, I'm at a liberal arts university where you're expected to know a little botany and zoology and ecology and molecular biology and everything. I freely confess I have weaknesses, but I work on it.
No,
Yeah.
not on our show. Yeah, uh the back to the museum, one of the talks or the one talk I participated in, we had each of the departments in the museum does like a talk, and so I was part of the and so I was in the mammalogy department, so we got to have our mammals at the museum, I think, because it's in evening. And so I actually had a difficult time being able to do it because I had a math test that was earlier that day, and so I had to reschedule that. Fun times. But I got to talk. I had a little stand, and I got to talk about the clade Atlantogenata. So I was telling people about xenarthrans and about
Okay.
Afrotherians. and it was very fun. I enjoyed the heck out of it. You know, uh, and we had one of the interesting things we had was we've got a lot of plasticine deposits here in Louisiana, and
Okay.
so we've got a ton of sloth stuff and mastodon and mammoth things, and so I had some mastodon teeth and I had some mammoth teeth, and I was uh, showing people the difference in the the teeth structure because mastodons were browsers; they were browse from the trees. Mammoths, on the other hand, had grazing teeth, uh, and so I think it's interesting because even though modern elephants, Loxodona and Elephas, although I think there's been some weird stuff going on with the Loxodonta group recently, so we'll see how long they retain that name. Uh, if determine like the two Loxodontas, like Cyclodus and Africanus, aren't actually closely related to each other.
Oh,
So
really?
they're descended from two different uh, uh, elephant species. Cyclodus is descended from Paleoloxodon, I think, and um, Africanus is descended from another Loxodonta species. I can't remember the name of off the top of my head, but yeah. So basically, it's these two independent lineages that have the same genus name, and so I'm not sure. I don't know if anyone's really getting on that right now, but that bothers me. And so anyway, so I think it's interesting that even though mammoths are more closely related to modern elephants, uh, the modern elephants have the teeth structure, I think, of mastodons. They're browsers, not grazers, at least for
Hmm.
the most part, which I thought was pretty interesting. That
Convergent
was
evolution. Yeah. Yeah, it
yeah.
happens.
But believe it or not, while we were there and I was talking to people, I had on my table is Dr. Esselstyn. He's the he's no, that wasn't him. It wasn't him. It was Mark, one of the geneticists. He tells me uh, you should have a phylogeny of mammalia or of placentalia, so that you can show the people how these are related to other mammals. And so they printed out a paper with different phylogenies, like ones that we've
Uh-huh.
and so this woman comes over. Uh-oh, you already know we're in trouble. There's a woman involved.
No.
And
No,
so
a
this so
Louisianan involved is what I'm worried about. Yeah.
there you go. Hunting. Okay. So what happens is she comes over and she has like 4 or 5 kids with her, I think, something like that. She looks at the paper. She sees the different phylogenies, and she says to me, she says, "Is this what they teach in the schools?" And I thought, well, I mean, your child probably won't learn specifically about Atlantogenata, but the general mammal
Yeah.
clade is discussed in biology class. Yes, she said. She like kind of leans in. She says, "Isn't this evolution?" I was like, "Oh no." At first, I didn't really know how to respond, and then she said, "Oh," and she said that, and I thought, "Oh no." And so, I just thought, really, come to the museum.
Maybe she was enthused because she was happy that you were teaching evolution to kids.
Uh,
Is
I
that a
don't think
possible? Oh, you
I
don't
don't think
think
so.
so?
I think um she said something like, "Is this a variant of is this taught at like most schools?" And I said, "Well, it is state curriculum, so yes, it's taught in the high school and college level. Wasn't taught for me in middle school, but I don't know about most schools in the United States. Louisiana is 49th in education. That's why we had the phrase, thank God for Mississippi,
Yeah.
right?"
I'm so sorry. Yeah.
It probably is in other states. Um,
Yeah.
I remember someone once said that you pretty much just have to teach 3 concepts, which is like heritability, variation, and time, or something like that. As long as you just get those concepts in their heads at an early level, it's a lot easier to teach evolution.
Right.
But But yeah, that was a fun night. We had some other really interesting museum groups, and this kind of comes back to the blood of the ice fish, or the herpetologist gave their talk. Actually, before I go there, Dr. Esselstyn, who's the head of the department, he gave his talk on the shrew rat he found in Indonesia, I believe, called that they named Posidentomes. It
Okay.
doesn't have incisors, or no, it doesn't have molars. It doesn't have any molars. It just has incisors.
Oh.
So they named it Posidentomes vermidax, which is few-toothed rat earthworm destroyer.
So that's what it lives on, is small invertebrates.
Yeah,
Yeah.
yeah, it is so specialized for these invertebrates that it's just going after those. So it lost its molars.
Yeah. Huh.
It's crazy. It's crazy stuff. But anyways, so the herpetologists, they gave their talk, and what they talked about um uh was, oh, I just got, so someone in my this guy Blake Bramley in my comments said hashtag not last as far as for our education. Um, so the herpetologist gave a talk about this. Uh, was it a skink he found? Or no, he didn't find it. He was doing research on it, called Prasinahema.
Uh-huh.
Yeah, the green blood. It has the green blood because it's a biliverdin, isn't it?
Oh, really?
Yeah,
I
it's
didn't know about this, but okay.
yeah, it has high levels of biliverdin, which would be like toxic, because that's like what causes
Yeah.
isn't it?
Yeah, biliverdin's not good for us. So I must have acquired some tolerance for it somehow. Hmm.
Yeah, it's even its bones are green.
Oh.
It's very weird.
Uh-huh. Yeah, biology's weird everywhere.
Yeah, it's very very bizarre, and so he was talking about that, and I got to see a lot of neat stuff, working at that museum. But
Uh-huh.
and so I guess so one of the things I wanted to ask you about was uh something I'm interested in is petamorphosis.
Uh-huh.
Can you talk a little bit about the petamorphosis in evolutionary history?
Yeah, I mean, there's a lot of work that's been done on that. Stephen Jay Gould's book, Ontogeny and Phylogeny, has a lot of good material on this whole process. And really, it's an aspect of a central issue in development, which is allometry. That what happens during development is differential growth of different tissues, different organs, limbs, things like that. And so, one of the cheap and easy ways that you can change the morphology of an organism is by changing the timing of its growth. So, either retain juvenile characteristics or accelerate growth and get, hyper adult sorts of characteristics. So just by playing with the timing, you can affect morphology in profound ways. So, there's a fair bit of work done on this in amphibians, because amphibians are a good model for this. It's also postulated that it plays a real role in human development, because adult humans look more like baby chimps than adult chimps.
Oh yeah, that was in Dawkins' book, The Greatest Show on Earth, I think.
Yeah, yeah, it's been known for a long time. And so one idea is that what happened in our evolution was we simply slowed down our rate of development of certain tissues. So we retain juvenile characteristics like a small face, small jaws. Think of all the baby-like things that make
Hmm.
babies cute, and it's things like that small face. The proportions change, so you can just play with these. Uh, there is a little bit of a problem in much of the literature with this, in that they tend to make these sweeping statements. where it's clearly not simply that you have wholesale acceleration or slowing of growth, because
Mhm.
you've got to have mechanisms where different tissues grow at different rates. So you get more complex morphological effects. I wouldn't say that we are simply neotenous chimpanzees, for instance. We've got a bunch of specialized things going on in development to give us these particular proportions and these particular facial structures. Uh, so it's got to be finely tuned. So it's a nice,
Uh-oh.
elegant explanation, but it's, you said oh because I said finely tuned, and that sets off all kinds of, yeah, finely tuned.
Wee woo, wee woo.
Natural processes only. No designers tuning things.
Well, yeah, that's that I watched. Yeah, I mentioned I watched some of your debates. You did the one with or something Jeffrey or Jeff. At one point, you guys were talking about giraffe evolution, and he said there were no intermediates with
Oh, right.
an intermediate neck or something like that.
That
What was
was
his name?
Jerry Bergman. Was this?
No, it wasn't the one with Jerry Bergman because I saw that one was he did the whole irreducible complexity thing.
Yeah,
This was
that
like on a
was.
radio or something, on a radio.
Oh, right. Now I remember. That was years ago. Yeah, that was a radio show, and he came on with this standard spiel that there are no intermediates, no transitional forms of any species on Earth, and he used as an example giraffes, and yet there are examples all over the place in
Mhm.
paleontology of, giraffes and camels. They're all related
Mhm.
with different degrees of neck development, and um, so it's like, he was the guy who was writing a whole book on fossils.
Oh, that's right. Like
Yeah,
billions of missing links or something.
yeah, and I named a few to him, and he didn't know what they were. It
That's
was,
oh
yeah,
boy,
yeah,
that's right.
I'm mainly a cell and molecular guy, so I'm not an expert on all the fossils out there, but so it's particularly bad when even I can trot out a few examples just from my casual knowledge of evolution, and this guy's pretending to write a whole book on it. Yeah, that guy, I can't remember his name.
It had
Uh,
Jeff in it. I know that. I
yeah,
can't remember the rest of it though.
and I also remember that he is an MD from Eugene, Oregon, where I
Oh
went to
boy.
school. So, yeah.
Yeah, I've met a few doctors with some wacky ideas. I
Uh-huh.
actually I kind of I joined a group of atheists on Twitter because they saw me sparring with this guy who's an actual MD anesthesiologist, but he didn't understand how mutations work. He was saying stuff like there are no de novo mutations and things like that. And he said that, and I sent him a paper on independent lineages of garter snakes developing de novo mutations to eat venomous Tarica, which is their it's a like a salamander, I think.
Yeah,
It's
yeah.
an independent lineage of garter snakes develop these de novo mutations, and so
It probably didn't sink in.
he said, "Well, I sent it to him, and he just kind of looked at me and said, oh boy, RJ Downer just sent us a message that says religion, a neotenous spandrel sustained as a scorched earth defense."
Sure, yeah.
I like that.
Uh-huh. Yeah, but that, that
What?
that actually leads into something I have troubles with, and that is evolution is a big, complex, difficult field. And even within evolution, you can find different degrees of knowledge about it. As I mentioned earlier on, I'm primarily a developmental biologist, and I came into evolutionary biology by way of development. And there's also the classical population geneticist. That's an even more traditional way to enter into the study of evolution. And we get conflicts within the field when there are people who, for instance, there's this move afoot to completely revolutionize all of evolutionary biology. And it's largely promoted by people who haven't mastered the traditional population genetics approach to evolution.
What is this movement called?
What do they call it? They go by different names.
I don't know. I don't think I've heard of that, but that sounds interesting. I'll have to
Yeah,
look into it. Maybe we can get
yeah,
RJ on that.
yeah. Maybe he'll remember the name of it. I tend
Oh,
to.
Jeffrey Simmons.
Jeffrey Simmons. Oh yeah, he's the guy. Yeah, okay.
Thank you, RJ.
Yeah, yeah. He was so annoying. Haven't heard of. I haven't heard from him since though.
I
But
think I should
yeah,
listen to these guys.
there's a number of groups that have been working to develop alternative approaches to evolutionary biology. And I don't think they're going to succeed unless they master the traditional aspects of evolutionary biology. It
Yeah.
takes hard work to get, you know, classical population genetics is largely statistical and mathematical.
Mm.
And it's really easy for somebody coming at it from a different angle, like developmental biology, and saying, okay, well, there's all this cool stuff, and you can't explain it with your numbers. And the thing is, all too often they can. And at least if they can't, for
Oh,
you to explain it, it has to be done through the same kind of mathematical approach.
Rational Mind is wondering if you're
Yeah,
referring to the extended evolutionary synthesis, because
right.
we mentioned that earlier. Is
Yeah,
that what you're referring to? Because yeah,
yes. I mean, there's interesting stuff going on there. There's some smart people working there. There's also some batty people.
it's like Massimo.
Piliucci, yes.
Yeah, that's right.
Yeah.
That's right.
He's a smart guy. He's primarily a philosopher at this point, which is a useful contribution. Uh, but at the same time, I just don't feel like it's tough to say because I feel like, hey, there's some really complicated, cool new stuff coming along, and we don't have adequate explanations for it yet. But it's kind of premature to say that you are revolutionizing evolutionary biology. You can't say that until you've fully pursued all the other strategies within evolutionary biology to explain it. And there are a lot of them. Uh, it's one of the things I'm struggling with right now because I'm going to be teaching evolution in the spring.
Oh
And it's
boy.
like, yeah, it means I've had to dust off my math and remember
I
all that stuff.
I actually made a video recently on the mathematics of population genetics. The focus of the video was Hardy-Weinberg equilibrium,
Mm-hmm.
and so it was very interesting. That's something I've sick to death of hearing about. I I mean I can spit it all out now because I've heard it at least 4 times. I
Yeah,
and in just by my end of my freshman year, I've heard it at least 4 times. It's just it's like some things like that. That and I can't really think of another one off the top of my head, but Hardy-Weinberg equilibrium is always one they hammer into us.
yeah, we do that our freshman year here too. Fortunately, our curriculum we've sort of divided it into two. So there's one part that's strictly cell and molecular. So we talk about cells, genetics, and evolution. And there's a second part that's evolution, uh, biodiversity, and things like that, population genetics. So I teach the first part. I don't have to teach them Hardy-Weinberg yet. They get that the
Hey.
second semester. They'll get that over and over again in the second semester.
But
Yeah.
yeah, it was a is it's fun the first 2 times you hear it. And I know I'm gonna hear it again because I'm only a sophomore now, so it'll be back.
Yeah, well, but if you take an evolutionary biology course, you might get it like the first week or two, and then boom, you're moving way past that. You're not you're done with Hardy-Weinberg. Hardy-Weinberg is a really simple model
Mhm.
for changes in frequency of alleles in the population, and there's so much more.
Yeah,
Yeah.
I think that is very interesting. It, yeah, the and that was kind of what made Mayer and the others popular, wasn't it? Was like statistical analyses of genetics and things like that. That
Oh,
was like early in 1900s. Because I know Dawkins talked a lot about statistics in the Selfish Gene, which
Yeah.
I read that like a year ago. Very interesting
Yeah.
book.
That's more the domain of people like Fisher.
Oh, okay. Oh, yeah, Fisher.
Right.
Yeah.
Yeah, but the thing is that the people who did who were responsible for the neo-Darwinian synthesis included people like Fisher, but Meyer was not trained in genetics at all, and his understanding of genetics was crude and horrible, and it's kind of embarrassing now when you read it. He was a bird person. He was more of a ethologist and systematicist than anything else. Uh, Meyer's not a good person to read for understanding evolution. I hate to say it. He's got he lived forever, which meant he got to write lots of books. But sometimes it feels like most of his books, what he's doing is mentioning this thing so that he can take credit for it.
Hey, you sociality. Hey.
Yeah, yeah. Meyer did it all. Yeah, according to Meyer. But
anyways, we've been talking for about an hour now. Would you like to end it here, or
we probably should wrap it up because my
okay?
wife is expecting me to fix her dinner. Yeah, I have jobs to do. But it's been fun. Do it
That'll be
again
interesting.
sometime.
Yes, thank you very much for coming on, and I agree. We will have to do it sometime again,
Yeah,
sometime in the
because
future.
we've only scratched the surface.
So much more that we can look at.
Yeah.
I'm also glad my headset didn't cut out on me this time. It has done that for the past few talks I've done.
Uh-oh.
I don't know why, but I got a new. Well, it wasn't this one. It was my other one, but hey,
Uh-huh.
it worked this time, so that's good.
Okay.
But thank you very much for talking today.
Thank you. Talk to you later.
All right, have a nice day, Dr. Meyer.
You too.